Okay, great. Thank you. Okay. My objective here today is to talk about these groups of leafless plants, which I'm going to refer to as holomycotrophs. So there are some people use the term full mycoheterotrophs. I think of that term as being a little bit ambiguous because, for example, all orchids are full mycoheterotrophs to begin with, but then most transition to being autotrophs. This term that Hannah Rasmussen introduced, holomycotrophs, I think captures what these leafless things are really doing. The thing that we often focus on, and the talks, many of the talks we've heard today, talking about holomycotrophs, is really their trophic function, their trophic position. The fungi, for example, let's see. Oh, here we go. The fungi, they parasitize, for example. We haven't really been talking about that today, but there's a fair amount known about that. And certainly changes in plastome genomes, we've heard a lot about that today. That's a really interesting thing. But I want to also highlight these systems as being interesting for the things that they're doing on the reproductive side and suggest that they provide, in the same way that they provide unique opportunities for looking at things like molecular evolution, they may provide some unique opportunities on the ecological and interaction side as well. Okay, holomycotrophs. About 675 species of these, the great majority are angiosperms. Most of them, 92%, are monocots, so very appropriately discussed in a meeting like this. And 49%, or about 328, are orchids. And so again, appropriate for such a meeting. And Craig and I estimated many years ago about 30 shifts or slightly greater, and that number seems to be persisting as a reasonable estimate for that. And so I think for those of us who have seen these organisms, especially when we've seen them live, we recognize them easily. They're reduced organisms in terms of their morphology. The leaves are present but reduced typically to sheaths, which is not unusual for a monocot, certainly. But the reduction also often extends to the flowers. And the expectation, or certainly the comments in the literature that you see about these things, is that they often are autogamous or selfing. You see a lot of language about that. And that could make sense, and is certainly true for some. Perhaps if in fact, oops, I'm doing the same thing that other people have been doing. It is hard, I agree. All right, okay. If in fact the trophic shift opens up new habitats for these organisms, like dark places, for example, maybe those are places where insects wouldn't be so likely to follow. So shifting to self-pollination, autogamy, might be a way of allowing them to persist. The problem we have at the moment is it's really difficult to estimate the number of autogamous species and understand really the reproductive biology of many of these, just because many of them are so rare and they're just understudied in the field, basically. So that it makes it very difficult to assess this in a direct way. Okay, so for example, the genus Gastrodia. Here's a little bit of range in floral morphology for Gastrodia. Here's Flexostyloides, which was named by Kenji Setsugu a few years ago. And this is clearly a cleistogamous species, so it's autogamous, it's also cleistogamous. Then we have something like Sesamoides in the middle, which is reported to be selfing, but also reported to be insect pollinated, so possibly facultatively autogamous. And then another species here that was named most recently of all of these, and has the most sort of showy flower I think I've seen in a Gastrodia. So it wouldn't be difficult to imagine that this species is actually focusing on insect pollination. Another thing that's interesting about the genus Gastrodia is that if you look at the numbers of what we think is out there, in 2010, Phil Cribb and some other authors estimated there were 22 species of Gastrodia. This year, if you look at Plants of the World online, it's 99 species of Gastrodia. So either this genus is actively evolving, or we're not very good at finding these in the field. They're rare, easily overlooked. So there's probably diversity out there that we do not know about, okay? So if we think about floral biology in terms of autogamy as an endpoint in the transition to leaflessness, we'd expect probably to see more autogamy in the ones that are highly heterotrophic, the ones that have kind of gone the farthest down this road. And we've seen with some of the work that Craig and Hannah have shown that, you know, even in Corallorhiza, which I'll be talking about, we have some species that are kind of in the middle. They're doing some photosynthesis, but not much. And if we use plastome size as a sort of proxy for this, we could kind of map this out. Conversely, then we might expect to see maybe more outcrossing in the species that are not so far down the road, closer to the autotrophic end of things. Unfortunately, again, the data are really incomplete enough for us to really do anything rigorous with this at this point, because there are so many species for which we just do not know, even in Gastrodia, for which we really don't know what the state is. Are they autogamous or not? So just to kind of lay it out and map a few of these on here for you, this is a sort of linearized plastome, just to have an idea of plastome size. Down at the low end, and then focusing here on orchids, we're just kind of focusing on orchids. I will come back and touch on Thismia later, but this is really going to focus on orchids at this point. Pagoniopsis schenkii is, I believe, the smallest known currently at about 14 kilobases for the plastome. It's autogamous, at least it's reported to be autogamous, although it's an open flower, so it's not cleistogamous. You could imagine there would be a chance for some outcrossing to go on here. Then we have things like the Gastrodias, which come in, a number of them, maybe 10 or 12, have now had their plastome sequenced, and they're around 30 kilobases. There's some variation there. And here, as I alluded to on the last slide, it's possibly a mix of selfing and insect pollination there. And then we get up toward the other end, in close to things that are actually autotrophic, still autotrophic plants. So we have things like Dipodium from East Asia. This is Dipodium roseum, which is about 141 kilobases and is reported to be insect pollinated. And then up here, the largest plastome, I think currently noted from a holomycotrophic orchid, which is Corallorhiza macrantha, which is probably insect pollinated as well. It's a maculated type, but with a larger flower, found in Mexico. Gerardo Salazar helped Craig and me find this one time, which was great. And so you see there is a sort of range. Maybe there's a correlation with degree of heterotrophy and pollination system. We cannot yet really rigorously test that. Okay. Just want to touch on the fungal parasitism side too, because I'm going to try to bring a couple of strands together at the end. So most holomycotrophs that are orchids are using ectomycorrhizal fungi that are typically Basidium mycota. For their fixed carbon. The specificity for these is usually believed to be quite narrow, and certainly for those that we've investigated in Corallorhiza, that's the case, maybe down to the level of a single species, although many of them still haven't been studied, things like Gastrodia, for example. And so this would be consistent with the prediction that in things that are highly parasitic, highly heterotrophic, dependent completely on a host, that you want to specialize in order to become efficient at obtaining nutrition from that victim in this case. Okay. I want to talk a bit about orchid floral deception here too, and pollination side, because that's really kind of what I want to focus on in a way. Orchids are pretty well known for the mutualistic relationships that they have with pollinators. There are some really Classic examples out there like Engracum sesquipedale, Darwin's orchid from Madagascar with the long proboscis, and the moth that was finally found that actually has that, or long spur rather, and the proboscis that matches it then. But it's a little surprising sometimes to think about these facts, and Jim Ackerman and some others have kind of brought out these numbers that up to half of orchids are actually probably deceit-pollinated and are not actually providing a direct reward such that it's truly a mutualistic relationship. This sort of deceit pollination has sometimes even been called flower parasitism by van der Singel, for example. And I think that's a legitimate way of looking at it, because the deceived insect in this case is not obtaining any kind of benefit, but it is coming at a cost. That service is coming at a cost to that deceived insect. Okay, Corallorhiza. You'll see there's a theme of Corallorhiza for me and my former students. I've been working with this genus for about 40 years now. Considering there are only 12 species, you might say, what's taking you so long? It's just that we keep finding new things and new approaches, new ways to look at variation. So it's just been very rewarding to continue to revisit this genus. So this is some of the diversity here. We've seen with Hannah's talk, we saw maculata this morning. This is bulbosa, Odontorhiza, Trifida up here, maculata on this side as well, and then the coralloid rhizome system. But for the rest of this, I really want to focus on the one in the middle, the one that Craig has focused on for a long time. This is Corallorhiza striata. Okay, species relationship. Again, a small genus, and we think we have some idea of the relationships of the species here. Basically, there are two sides to the genus, two sister clades. There's what we can call the maculata side, and there's a reason for viewing it in that way, in that the flowers have a sort of similar plan across these species. And they also have a similar plan between striata and its related species too. And I kind of want to contrast those for you now as I tell a little bit of story about striata. Okay, so this is Corallorhiza striata, one of the showier species of Corallorhiza, distributed across the northern portion of the US and southern Canada, and then also in the southwestern portion of the US and down into Mexico and actually into Guatemala as well. Okay. If you look at striata, as with many species that are widely distributed, there's actually a certain amount of variation. And so there is that in striata. For example, we have this smaller flowered form from the southwest and into Mexico called Vreelandii. This is the larger flowered form, variety striata. And then two others. I had once referred to this as a variety. We're now viewing it as a species, involuta, which is Mexican. And then this species, which I described from Eastern North America 20 years ago, which is also a very small flowered species. But for the rest of this story, I really want to focus on this large flowered plant. And to just contrast the basic floral morphologies between these two sides of the genus now, using maculata and striata as our example. So for example, the labellum in these two, rather different in morphology in the sense that it's a rather flat labellum here. The margin is just flat. Striata is a very different thing going on. The labellum is thickened and upturned into a sort of boat-shaped or navicular sort of morphology. At the base of the labellum, we typically have a couple of ridges or lamellae. They're free on the maculata side. They're thickened and fused on the striata side. Looking from the side, this labellum is distinctly, obviously it's hard to see here without motion, but it's distinctly hinged and able to move very freely and kind of hangs downward. And this species, striata, also does not have a spur, A spur or small mentum, which we do see, or a mentum rather, or small spur, which we do see on the other side, which kind of highlights the idea that this doesn't actually produce any nectar. Okay. Even things like the coloration and spotting are quite different. So obviously, on the right side here, this striped morphology is very pronounced in striata. We don't see that in the other species. But one other thing that's really interesting is at the base of the column, we see various kinds of spotting on maculata and the other groups, whereas in striata, there's always a spot right there. There's a prominent spot. As far as we've seen anyway, there's always a spot right there. It's quite prominent, quite distinct. And we think it has something to do with the pollination biology. So even when we look across happens to be a little population sample with 5 individuals, 2 flowers from each, just laid out so you can see them. They vary in base color, in a sense, from very dark to brighter, but they all have that spot at the base of little bellum. Okay. What pollinates this? Oh, I hope I can do this. Let me show you. Let's see if this just, oh yes, watch. Curved abdomen at this point. Flies off. Okay. That's what this is. This is an ichneumonid wasp. The observations that have been made for pollination and the rest of the genus all involve small flies or halictid bees, sort of sweat bees. This is the only thing that we've seen that's pollinated by an ichneumonid. And ichneumonid pollination is rare in general, certainly for orchids. Ichneumonid do visit flowers in search of nectar, but the thing here is that we're always seeing males. It's exclusively males. And this species, Pimpla, it's called Pimpla pedalis, males. And here you can see a pollinarium attached to the thorax of this little guy. Okay. If I compare the distributions of the, of the plant and the wasp here, this is what we see. They're quite similar, actually. So you can imagine it could be an effective pollinator across the range. And we also have had no other pollinators reported. We've seen no other ones ourselves. So why does Pimpla pedalis come to this flower? It's a distinct floral morphology, different from the other species of Corallorhiza. It doesn't produce any nectar. It's only males that are visiting. And there's this odd abdominal behavior on the visit of the wasp. So what we think is going on is pseudocopulation, this specialized type of deceit pollination that occurs almost only in the orchids, just an example or two in iridaceae and in asteraceae, but all the rest are in orchids. Typically from Australian orchids, not sure quite why that is, a few examples from Europe and South America also, although depending upon how the pleurothalidae story works out, there could be many more in pleurothalidae that are doing this as well. And it's also mainly in Orchidoideae, rarely in the big subfamily Epidendroideae like we're seeing here. But this is the first known example of this from North America, certainly, and the second known example of this kind of strategy to use an ichneumonid. Okay. Typically with this kind of deceit, the plant will produce a pheromone mimic as well in order to bring in the male wasps. And so we've started to look at the fragrance compounds. And you can see here, this is a gas chromatograph, total ion chromatogram. And what you see here are a couple of compounds I highlight in particular, butyl hexadecanoate and butyl octadecanoate. This one has actually been identified as an ichneumonid pheromone before, although not that many ichneumonid have actually been examined for their pheromone, for the pheromones they produce. Okay. Just to return for a moment to this slide that Craig showed earlier, just to emphasize the narrowness of the of the specificity. So this is the range of Corallorhiza striata, really focusing on probably one species, Tomantella fuscocinerea, patagonica. We don't know if that's distinct or not. The black rhizomes that we see in striata are indicative of Phyllopheraceae, so it's a high degree of specificity. So our argument here is basically that this is a situation that we could refer to as double parasitism. So it's parasitic on the vegetative side on fungi, on the floral side on wasps. About 20 years ago, Martin Vidartando commented in the literature that we would never expect to see this, because the idea that you would be focusing on two sort of deceptive situations independently would be, as he put it, too evolutionarily unstable, by which I think he meant too risky in a sense, because if you failed on either side, you would be lost. But I think that's exactly what we have here. There are other systems of parasites, trematodes, for example, that utilize more than one host, but it's never in the same life stage. There are things certainly that have up to 4 successive hosts among the trematodes, but this is one where it's doing it in 2 at the same time. And one might think 4 successive hosts is also risky behavior, but it does seem to work for those taxa, and it does seem to be working for Corallorhiza striata. The question is, are there other places we see this? And I think kind of there are, and I just want to highlight those for you. So Gastrodia, Martos et al. In 2015 noted floral deception of Drosophilid flies in one of the species of Gastrodia. In Thismia, Guo et al., 2019, demonstrated that it's probably a single species of Glomeromycota here, that it's tracking on the vegetative side, and possibly a single species of fungal gnat that it's using for deceptive pollination of its flower as well. And then finally, the genus Danxiaorchis, which now has 3 species, which is unusual. It's in calypsone with Corallorhiza, very unusual in the color of the plant, bright yellow. Nothing else is bright yellow in calypsone. So it's not hard to believe that it would be tracking in on Lysimachia flowers. They've demonstrated that Yangii, this species, is a deceit mimic of Lysimachia pulling in halictid bees, and it might be true for the other 2 species of Danxiaorchis as well. They haven't looked yet. Okay. So my argument here, or our argument here, is that holomycotrophs provide this really interesting opportunity to look at this heretofore predicted to be kind of impossible situation. We seem to be seeing it, though. It seems to exist, given on this both trophic side and reproductive sides, this sort of parasitism. So maybe it's really not as unusual or impossible, certainly, as Martin Vidartando predicted. And maybe it's also suggesting that such parasitisms are more stable than expected. Maybe most of them don't persist for a long time, but they do persist for a while, at least long enough for us to be able to capture situations where they're doing both of these things at the same time.
Excuse me, so sorry. Time is done.
Right, and I think this is just to say, this is my last slide. We've seen a slide with a picture with a wasp on bentleyi, so maybe happening there, and we need to know what happens in involuta. All right, thank you very much.
Yeah. Thank you. Time for questions. Yep.
Let me give you this.
Yes.
Well, you kind of answered my question, but I'm going to ask it anyway. It looked like bentleyi and involuta have very small reduced flowers, and I wondered if they might be autogamous, but you don't have that. So.
Well, actually, that's a good question, Sean. Bentleyi, when I described that from a plant that looked a lot like that, the flowers did look like they were essentially almost cleistogamous. Subsequently, additional populations is now known from maybe 13, something like that population. Additional populations were found that had a more chasmogamous flower, but we still think there may be autogamy going on there. May well be true for involuta too, but maybe they're still producing the compounds or something, and yeah, we don't know why it's visiting. We tried to catch one, and we didn't see any pollinators. Tom?
One other question.
Do we have time for that?
Okay, I'll just,
Okay,
yeah.
just a minute first. This one.
Oh.
Hi, I must say I really enjoyed the slide where you showed the morphological variation in striata. I was wondering if those flowers came from the same individual, same population, or different populations, because those were quite a range of flower colors.
You mean the one where it had 10
Yes,
flowers?
yeah.
One population. I chose from the population as much variation as I could find to illustrate the extremes of variation. So one population.
Thanks. So one suggestion. Often there's not a black and white difference between autogamy and outcrossing. And so
Right.
There's a continuum.
Even in Corallorhiza.
Exactly. And so I'm wondering whether it might be helpful, whether it could be done, to relate measures of outcrossing, say length of petals or population genetic measures, to establish a gradient that way. Have people done population genetic studies on any of these things to look at what the inbreeding coefficient is?
Almost nothing. I'm not really aware of anything. Are you, Craig? Well, I guess you have done some. That's right. You have done some, but outside of our little piece of Corallorhiza, I don't think anybody's looked at all. Yeah, that's Craig is focused on with bentleyi and stuff is looked actually, but beyond that, it's not. It's just known from that one little piece, so it's hard to relate it broadly, I guess.
Yeah, you brought up the issue of having double parasitism. You know, I really don't have any problem with that because you can say the same thing for specificity, and both specificity in below ground as well as above ground in pollinator relationships, and the vast majority of orchids do both. They're highly specific. The median is one pollinator, and I think the mean is 3. And there's some outrageous outliers that have gazillion pollinators that skew it. So it's kind of a trait of the orchids, and yeah. So I wouldn't worry about it.
Okay.
Thank you so
Are
much.
we finished? Thanks.
Yeah.